Hello and welcome to EaseGenetics. I'm Dr. Kaveh, and I'm here to take you one step further in understanding genetic linkage. In today's video, Linkage 201, 3-point gene mapping, we'll delve into the process of mapping the location of 3 genes on a chromosome. Let's get started. The topics we will discuss today are as follows. Determine the linkage between 3 genes, effect of double crossing over between 3 genes, determine the parental genotypes, determine the double crossing over genotypes, determine gene order, calculate recombination frequencies, construct the genetic map, and interference and coefficient of coincidence. As I mentioned in my previous video, Linkage 101, the number of crossovers between 2 genes must be odd to produce recombinant gametes in the offspring. When 2 or any even number of crossovers occur between the 2 genes, all the gametes will be of parental types. But if a third gene, such as D, exists between the 2 crossover events, not only does it break the distance between the 2 genes if they are even more than 50 map units apart and show them as linked genes in recombinant gametes of the offspring, but it will also appear among the recombinant gametes resulting from double crossovers. This is very important when examining 3 genes to determine their order on the chromosome. Also, the gene between the 2 crossover actually breaks it into 2 single crossovers. Just as the independent and linked states of 2 genes can be compared, where we saw that the only difference between them is the frequency of the gametes, the same comparison can be made for the case of 3 genes. Here we see the 8 types of gametes produced by a trihybrid organism. If the genes are independent, the frequency of these gametes will be equal. Now, if the 3 genes are linked, we will see 4 scenarios during meiosis, here labeled as cases 1 to 4. For simplicity, I have considered all 3 genes in the cis configuration. In the first case, no crossover occurs, resulting in all gametes being of the parental type. In the second case, a single crossover occurs in some cells between genes A and D. As a result, recombinant gametes for these 2 genes are seen along with parental types. In the third case, a single crossover occurs in some cells between genes D and B, leading to recombinant gametes for these 2 genes and parental types as well. In the fourth case, a double crossover occurs, producing both parental and recombinant gametes. The important point here is that parental type gametes are the most frequent. The frequency of recombinant gametes resulting from a single crossover in cases 2 and 3 depends on the distance between the genes. Additionally, since the probability of 2 simultaneous crossovers, one between genes A and D and the other between genes D and B, is very low, the frequency of gametes resulting from double crossovers is the lowest. This observation is crucial for identifying offspring resulting from double crossovers because the middle gene is displaced in such offspring, which is the key point. By comparing these gametes with the parental types, we can determine the correct order of genes on the chromosome. Here, I have categorized and organized the gametes resulting from the 4 scenarios for better clarity. As you can see, in the first case, we observe parental gametes. In the second case, we see gametes resulting from a single crossover between genes A and D. In the third case, gametes resulting from a single crossover between genes D and B are observed. And finally, in the fourth case, we see gametes resulting from a double crossover. The important point I want to draw your attention to is that within each group, the pairs of gametes are exact opposites of each other in terms of the arrangement of dominant and recessive alleles. Let's call these reverse pairs. This will significantly simplify our work as you see. So, when we are dealing with three linked genes, there are two very important points that we must always remember. Number one, only the middle gene is changed in double crossing over gametotypes. And number two, the frequency of these gametes is the lowest. Here you see a simple example that shows why the frequency of offspring resulting from double crossovers between three genes is the lowest among all offspring. Let's assume this is our genetic map. The distance between genes A and D is 30 map units, and between genes D and B is 20 map units. For a double crossover to occur, crossovers must happen simultaneously in both regions 1 between A and D and region 2 between D and B. In fact, we can say that one crossover must occur in region 1 and another crossover must occur in region 2. As I emphasize on the word and here, in probability, whenever we calculate the likelihood of two events happening simultaneously, event 1 and event 2, we must multiply their probabilities. So the probability of both events happening is 30% multiplied by 20%, which equals 6%. So as you see, the probability of a double crossover is significantly lower than that of single crossovers. Now let's examine a basic example based on the information we've discussed. Let's consider three traits in fruit flies: wing length, eye color, and leg length. Long wings, red eyes, and long legs are dominant to dumpy wings, purple eyes, and short legs, respectively. For the recessive alleles, we'll use the lowercase letters D, P, and S respectively. And to show the dominant alleles for each trait, we'll add an uppercase plus. Let's say at this stage we don't know if these three traits are independent or linked. Now we need to perform a cross. We'll test cross a trihybrid fruit fly. This means we'll cross a tree hybrid with a fruit fly that has the recessive phenotype. And we already know the genotype of this recessive fly. It is homozygous recessive for all three traits. If these are the results, as you see, all 8 possible phenotypes from a tree hybrid test cross appear among the offspring. Now, let's take a look at the numbers of offsprings for each phenotype. Out of 800 offspring produced, approximately 100 were obtained for each phenotype, which aligns with the expected ratio if the genes are independent. Some phenotypes have slightly higher or lower numbers. A chi-squared test confirms that the differences are not statistically significant, supporting the conclusion that the three genes in question are independent. But what if we get these results? While all 8 phenotypes still appear, the number of offspring in each phenotype is no longer equal. There are significant differences among them that are even visually noticeable, and a chi-square test would show a statistically significant deviation from the expected ratios under the assumption of independent genes. From this, we can conclude that the three genes are linked. Take a look at the 2 phenotypes that have the highest numbers. These are the parental types, while the 2 with the lowest numbers are the double crossover types. The remaining 4 phenotypes correspond to the 2 single crossovers. As we know, all offspring receive one chromosome from the test cross parent carrying recessive alleles for all 3 traits. By examining the offspring's phenotype, we can determine whether they inherited a dominant or recessive allele for each trait from the tree hybrid parent. For instance, phenotypes 1 and 8, representing the parental types, show that the arrangement of all 3 genes on the parental chromosomes is in the cis configuration. So as you see, we can write the gametes genotype received from the trihybrid parent for each offspring. As I previously mentioned, the 8 phenotypes can be grouped into 4 reverse pair types for easier analysis. Group 1 is the parental type with the highest frequency, while Group 4 is the double crossover type due to its lowest frequency. Groups 2 and 3 represent single crossovers. However, as we have yet not determined which gene is in the middle, we don't know between which genes single crossovers happen in groups 2 and 3. Identifying the middle gene is the next step and always my favorite. First, on the left, look at the parental type chromosomes at the top and one of the double crossover chromosomes below them. As we know, the middle gene switches places during a double crossover, while the flanking genes remain unchanged. By comparing the double crossover chromosome with the parental chromosomes, you'll notice that it most closely resembles parental chromosome 1, with the only difference being in gene S. This identifies S as the middle gene. Now on the right, if we compare the other double crossover chromosome, it most closely resembles parental chromosome 2, and again S is the only differing gene. After determining the middle gene, we rearrange the parental type chromosomes to place the gene S in the middle. To construct a genetic map, we need to calculate the distance between genes D and S, and genes S and P. Let's identify the single crossover regions for groups 2 and 3. Here, the parental chromosomes are shown with the corrected gene order. By tracing the path of a chosen offspring chromosome in group 2, for chromosome I is the blue and for chromosome II is the red path, we observe that a crossover occurred between genes D and S. This confirms that group 2 corresponds to single crossovers between genes D and S. Using the same method, we analyze group 3 offspring chromosomes to confirm a crossover between genes S and P. By comparing the offspring chromosomes with the parental chromosomes, we trace the paths and identify the crossover points. Here, the path for chromosome I is in blue and for chromosome II is in red. With this analysis, up to now, we've determined the middle gene, the correct gene order, and the locations of single crossovers. Now we can calculate the genetic distances. To calculate the genetic distance between genes D and S, we add the number of single crossovers between these two genes to the double crossover offspring, divide by the total number of offspring, and multiply by 100. In this example, the frequency of crossovers is 13.75%, equating to a distance of 13.75 map units or centimorgan. Similarly, the genetic distance between genes S and P is calculated by summing the single and double crossover frequencies for this region, dividing by the total number of offspring, and multiplying by 100. Here, the distance is 26.25 map units or centimorgan. With the gene order and distances established, we can construct the genetic map. We can draw a hypothetical chromosome segment marking the 3 genes in their correct order and relative distances as you see in this slide. Next, we explore interference and the coefficient of coincidence. Interference refers to the reduction in the likelihood of a second crossover event in a nearby region after one crossover has occurred. It reflects the non-random distribution of crossovers during meiosis. The interference I is calculated as I equals 1 minus C, where C is the coefficient of coincidence. Let's see how it looks like. In this image, we see a portion of two sister chromatids with two crossovers. The meaning of interference here is that due to the physical properties of chromosomes, when a crossover occurs in one region, the chromatids need a certain distance to bend and perform a second crossover. This distance is 5 map units or centimorgans. So, if a gene is located anywhere within this region between the two crossovers will occur outside this region. Earlier in this video, we calculated the maximum amount of crossover between two genes that were 30 and 20 map units apart, and we saw that the maximum possible double crossovers were 6%, which is our expected value. However, in reality, this does not happen due to interference, and the frequency of offspring resulting from double crossovers is always lower because of the 5 map unit region around the middle gene. We can't directly calculate the interference, but we can calculate the coefficient of coincidence. Coefficient of coincidence measures the actual frequency of double crossovers in a given interval relative to the expected frequency if crossovers were independent events. It helps to evaluate the extent to which crossovers deviate from randomness. This means that by knowing the distance between the three genes and the number of observed double crossover offspring, we can calculate the coefficient of coincidence. Then, using the formula, we can determine the amount of interference. The coefficient of coincidence is calculated as the ratio of observed to expected double crossover frequencies. For the observed frequency, divide the number of double crossover offspring by the total offspring. For the expected frequency, multiply the frequencies of the two single crossover regions. In our example, as you see the calculations, the observed frequency is 0 and 125 10,000ths, while the expected frequency is 0 and 36 thousandths. Thus, the coefficient of coincidence is 0 and 347 thousandths, meaning only 34.7% of the expected double crossovers occurred. Before we calculate the interference, let's explain what 34.7% means. It means that in reality, we only observed 34.7% of the double crossovers that should have occurred in the example. In other words, there should have been about 29 double crossover offspring in the example, but we only observed 10 out of 29. This means that we did not observe 19, which is the amount of interference that will be calculated in the next slide. Finally, interference is calculated as I equals 1 minus C. In this example, interference is 0 and 653 thousandths or 65.3%. This concludes our discussion on 3-point genetic mapping. Thank you for watching. I hope this video helped you understand 3-point gene mapping. If you found it helpful or enjoyed it, please give it a thumbs up. It really supports the channel and helps others discover this content. If you want to see more videos like this and keep learning about genetics, don't forget to hit that subscribe button and turn on the notification bell so you never miss an upload. Feel free to share your thoughts, questions, or suggestions in the comments below. Thanks again for your support, and I'll see you in the next video. Take care.